with what really got me going on having to do something. And this is the question that quality control starts with clinical quality requirements. How much error is allowed? This is the definition that I was brought up with 25 years ago. The amount of error that can be tolerated without invalidating the medical usefulness of the analytical result. Makes sense. The result has to be good enough to be medically reliable to the patient. So, whoops. What happens is that the concept is you have a true value, you have a medically useful range. Beyond that range, results are invalid. They're no good. They're not good enough to lead the doctor to the right diagnosis, and they're harmful to the patient. That led to the statistical definition, which is a statement such as, or that morphed into, this result is expected to be within X% of the true result, Y% of the time. X is the performance standards, etc. Then we had the regulatory definition. You have to pass the regulatory inspection. Neither of these really mention patient care. We have biological variation, which is based on patient criteria. Mosby's medical dictionary says they sort of merged the two. The amount of error that can be tolerated without invalidating the medical usefulness of the analytical result. Allowable error has a 95% limit of analytical error. Only one sample in 20 can have an error greater than this limit. Does it make sense to you that one result in 20 can be medically invalid? Most people would say sometimes, but certainly not all the time, that one result in 20 can be medically invalid. I'd say no, it makes no sense whatsoever to me. So this week's online discussion is, do you think it is okay for one result to be medically invalid? And then assume for the moment that this change I'm talking about will make a significant improvement. What are the obstacles we need to overcome? So there are driving forces that are taking us forward, and there are restraining forces that are holding us back. The reasons to move forward, it's recommended. So what I'm talking about is setting allowable error limits, clinical allowable error limits, and varying QC rules to meet the current performance of your test. This is recommended by experts. It's proven to reduce the risk of patients receiving bad results. It makes sense to use more QC for poorer methods and less QC for good methods. You reduce lab costs and the time for false positives, and you reduce clinical costs by eliminating the bad results. Those clinical costs are something that is quite often neglected. We talk about the cost to control quality, but we often forget what are the costs of failure to control quality. And finally, there was a QC check. I opened up the microphones, and the first comment was, we haven't seen anything different since the very first poll question. You mean everyone was stuck watching one slide? Oh no, what can we do now? But every problem creates an opportunity for improvement, an OFI. And most quality problems create an opportunity for education. So this leads us to an unexpected quality lesson. A webinar with no images fails quality standards. 50% of it was bad. Would any lab consider method performance acceptable if 50% of patient results were bad? Some do. In a recent survey, I asked people, when would you stop reporting? 60% said, we don't have any defined directions of when to stop. Well, if you don't stop and clinically significant errors are reported, then doctors make the wrong decisions, patients get wrong or delayed diagnosis or treatment, the costs and the risk and the patient harm goes up dramatically. Another full 20% said we stop if sigma equals 0. Well, guess what, folks? This is 6 sigma. There are 6 SDs from where the mean is now to where it would be if the method failed. This is 4 sigma. There are 4 SDs between the mean and the allowable error limit. This is 2 sigma, and this is 0 sigma. Sigma equals 0 when 50% of patient results are bad. It is unacceptable for a webinar to be 50% bad. How many bad patient results are acceptable? And the cold hard truth is that many labs do not set clinical quality standards. Most labs do not yet define when to stop. A lot of labs do not stop until 50% of results are bad. And most of the labs that do set quality standards follow the statistical limits, and they do not stop unless 5% of the results are bad. One single test will revolutionize lab quality control. Please keep asking yourself, does it make sense? The last of the restraining forces were people saying, I don't need TEA limits to control quality. It's too difficult to set TEA limits. It's fine to use the same QC rules for all the tests. It would confuse staff too much to change. We don't need to do this to pass inspections, and our software won't handle many rules. Those are the questions we need to overcome. The way these work is I talk for 30 minutes. What doesn't get said this week goes on to next week. These are unstructured, informal. It's a discussion.